welcome. Thank you for joining us for today's episode of Data for the People, presented by Rock EDU Science Outreach. My name is Devin Collins, and I'm Program Manager for Outreach and Engagement at Rock EDU. Data for the People is an interactive web series that invites all people to engage with relevant scientific research topics presented by scientists from our own communities. Information in these presentations is sourced directly from primary scientific literature, and it's explored in a way that maintains the rigor of the research while eliminating inaccessible jargon. Because everyone has a right to primary scientific information and to explore how science connects with our society. The theme for this season of Data for the People is People in Medicine. During this season, we have explored the science of how we study ourselves. We've learned how our complexity as organisms and as people with unique lives, identities, and characteristics can create challenges and opportunities for improving human health. We've examined how historical biases and power structures have influenced the way that scientific knowledge is generated, valued, and transformed into medical advances. We've also considered topics like the ethics of genome sequencing studies, gender and race-balanced clinical trials, and the role of social inequities in healthcare access and outcomes. And finally, we've explored how new and innovative practices, from designing experiments to better model human biology, to uniting patients and scientists in the study of rare diseases, has allowed us to answer another key question: how do we get better at studying ourselves? And so we're going to explore that through a very exciting topic today. Diseases can be very complex. And in many cases, what sounds like one single disease may actually be better described as a collection of many different diseases, each with its own underlying causes, characteristics, and importantly, avenues for treatment. Cancer is a very good example of this. And although all cancers share a key number of hallmarks, like abnormal growth of cells for one, also the signs and symptoms of cancer can differ depending on which organ or part of the body is affected. And the specific cells involved, the specific causes can also, that led to the cancer in the first place, can also change what we see in the clinic. So today's speaker will give us some insights into that complexity and how scientists can sort of tease out some of that complexity of cancer's origins. Our speaker today is Jude, who is a PhD student at Weill Cornell Medicine. Jude focuses on understanding the molecules and genes that are involved in the development and progression of prostate cancer. To do this work, he uses a number of techniques like in vitro studies, tissue studies, and drug response studies, among others. And before graduate school, Jude completed his undergraduate education in Boston and worked as a research technician at various institutions. In his spare time, he volunteers in outreach-related activities, runs a podcast dedicated to highlighting the journeys of aspiring physicians and scientists. I myself just subscribed when he told me about it, so you should do the same. And he also practices martial arts and plays guitar in his free time. So before we get started with Jude's presentation, I just want to go through a couple of acknowledgements and norms. First of all, I have a disclaimer for you. We come from a background of scientific research. We are not medical doctors, and we won't be giving medical advice today. This presentation is explicitly for the purposes of education and discussion. Second, we don't want to go outside the bounds of our knowledge. So as you are submitting questions, we ask that you only ask questions that are related to the topic being presented today. Third, to help keep everything organized and while allowing audience members to ask questions, we will open up the Q&A after the speaker is finished presenting the research. We'll try to take as many questions as we can, and thank you so much for being with us on this. And then finally, these sessions are recorded and archives will be shared at rockedu.rockefeller.edu, also known as Rock EDU Online. So A quick reminder, I know I just said this, but as Jude presents, be sure to plug your questions into the Q&A box at the bottom of your screen. We'll take time after the presentation to get through as many of those questions as possible. And with that, I am going to stop talking. Jude, thanks so much for being with us today. I'm going to turn it over to you. Oh, Jude, you're muted.
All right, thank you, Devin, and good evening, everyone. It's great to present to you today. I'm super excited for this. Okay, so the title of my talk is, as you guys can see, is the stromal cells in prostate cancer pathobiology, friends or foes. But before I get into everything prostate cancer related, I want to tell you guys about myself. So I was born in London in the United Kingdom to Ghanaian parents. My parents actually met coincidentally in London and had me. And a few months later, I moved back to Ghana, went through my entire education in Ghana. So essentially, I was raised in Ghana, in the capital city of Accra. And I immigrated to the United States in 2014 to pursue my higher education and realize my career in science. I attended the University of Massachusetts Boston, or UMass Boston, where I discovered my love for prostate cancer research. And this came as a result of the stark prevalence rates of prostate cancer and mortality in Ghana. I would say it's basically a death sentence if you have cancer in Ghana. And I acknowledged this early on and decided to contribute like my time towards addressing this issue. Okay. So as I said, I went to UMass Boston, and I got into this program known as the UMass Boston Dana-Farber Harvard Cancer Center. If you don't know what Dana-Farber is, it's one of the top premier cancer research institutions in the United States. And I got into this program in my sophomore year, actually, in the summer. And I would say this was really the catalyst to realizing, to kickstarting my career in translational science work. As a result of the years I spent in the lab of Dr. Chang-Meng Kai and a few other opportunities afterwards, I developed a set, I'll say a great repertoire of skill sets, which are highly transferable and culminated in these publications that you can see at the bottom. So publications are really the end goal of a research question. And that is like a milestone that every single researcher undertakes in his or her career. Another win I want to highlight is an award I received, specifically being third place in the Ruth and Silene MD Award at the Harvard Medical School New England Science Symposium. And fun fact, I actually won this award on my birthday, which was great, which felt really good. Okay. So fast forward to 2023, I'm a grad student, specifically a PhD candidate in the lab of Christopher Barberi, where I study the interplay between transcription factors in prostate, in starting or kickstarting prostate cancer. So here's my team right here on the left. Every single, I would say I adore every single individual and I love working with them. And here's my boss, who is an awesome and such a caring individual in so many ways. And on the right, you can see me in my elements. This photo was actually taken during a retreat last year at Mohonk Mountain, which is upstate in New York State. So when I'm not pipetting or adding or taking something out of a cell, I am you can find me in the studio downtown on the west side, just punching stuff, beating stuff up. Yeah, I just want to show you guys what I do every day. I also run a podcast focused on highlighting the stories and journeys of underrepresented aspiring physicians and scientists. We currently have 4 episodes out so far, and I would love if you guys can follow us on Instagram by by clicking this QR code, or by taking a photo of this QR code, sorry. And the last venture I want to highlight is one that's near and dear to my heart, known as the Path to Science Initiative, a URM's journey, or SciURM for short. And I co-founded this venture with my PhD classmates, Winnie Ciso and Stephen Ruiz. And the aims of our organization is to expose the works of URM scientists and provide mentorship and research opportunities for students at the undergraduate and high school levels. So now I'm going to get into my paper, which talks about the contribution of the resident cell types in the prostate to prostate tumorigenesis, which basically means prostate cancer initiation. So in this talk, we're going to focus on we're going to start really from the ground up. So I'm going to take you guys through conception, really, when life is formed. I'm going to start from that point, talk about how the prostate organ is formed, the types of processes which regulate its function, the ways in which prostate cancer can happen, and how we study prostate cancer, and new tools to actually improve prostate cancer detection and diagnostic approaches. Okay, so I want to start off with the hard facts. So according to the American Cancer Society, in 2023, there's going to be a predicted 300,000 new cases of prostate cancer in the country, and approximately 35,000 deaths, which basically confers the second highest leading cause of cancer death in men, right behind lung cancer. And this sort of signifies the importance of studying this field, because it does kill a lot of people, sadly, a lot of men, sadly. So as I said, we're starting from the ground up, and here on the left side, I have an embryo, which is the result of a male sperm and a female egg fusing, forming a zygote, which undergoes a number of differentiation processes or splitting processes to begin the process of life. And shortly after fusion of the sperm and egg, there is a number of replicative or differentiation processes, which leads to the formation of 3 distinct germ layers, known as the ectoderm, the mesoderm, and the endoderm. So these 3 germ layers give rise to all tissue types in the body. And I am going to use the male reproductive system as one of the systems, as an example of the mature organ system, which arises from these 3 germ layers. So in the case of the male reproductive system, the ectoderm forms the skin of the sex organs, as you can see here. The mesoderm forms the connective tissue that we find in sex organs. And the endoderm forms the inner organs of the inside organs. So basically in the context of the male reproductive system, I'm talking about the bladder and the prostate. So looking deeper into how the prostate organ is formed, I want to talk to you guys about the different cell types which we can find in this prostate organ, and how they talk to each other, communicate between each other to induce organogenesis, which means the formation of organs, and how their communication is intercepted or disrupted to lead to prostate cancer. So here at the top, I have a timeline of prostate maturation from the fetal stage to the adult stage. And as I said, this prostate organ consists of different cell types which talk to each other to regulate normal development and homeostasis. So as you can see here, this image right here is histological slide, and this is basically a tissue section of the prostate, and it's stained with 2 different dyes, which basically annotates the resident cell types in the prostate. So you can see two colors here, pink and purple. The pink color represents the stromal cells, which transmits growth factor signals and a signal known as the androgen receptor signal. And I want to highlight the androgen receptor a little bit. The androgen receptor is actually the master molecule of the prostate. It is responsible for maturation of the prostate and normal function of the prostate because it binds to hormones. It binds to hormones, specifically testosterone. So stromal cells transmit these signals to epithelial cells, which are highlighted in purple right here. And this communication between stromal cells and epithelial cells confers normal functioning of the prostate organ. And before I forget, actually, the normal function of the prostate organ is to induce secondary sexual male characteristics. That means a deeper voice, a broad chest, hairy skin. These are all male-oriented characteristics which are regulated by the prostate, by the processes that happen in the prostate. So here on the top right, you can see a prostate luminal cell, which is one type of cell found in the prostate where normal androgen receptor signaling happens. So during the process we call life, there are a number of events or phenomena which can disrupt the communication between cells and lead to the onset of cancer. Some of these are, we can classify some of these into the following groups: environmental, chemical, and genetic. Some examples of environmental cancer-causing agents or mutagens are asbestos found in cement. And you guys might probably have heard of asbestos from, if you watch TV a little bit. There's always ads on mesothelioma, which is caused by asbestos, actually. And another example of an environmental mutagen or cancer-causing agent is BPA, which used to be found in plastics. But upon this realization, BPA is being gradually removed from plastic manufacturing. Another class of cancer-causing agents is chemical, and we all know what tobacco does, which you should know is bad for you. And the last cancer-causing agent group I want to talk about is genetic. And some classifiers of genetic mutations which induce cancer are damage to genes which repair the DNA, and loss of genes which sort of like guard the DNA. And a prime example of that is a protein called TP53, which is right here. And as TP53, fun fact, is actually the most common mutation in cancer, like in all cancers. Okay. So summing up all the different cell types, I would like you guys to appreciate the spatial organization of the different cell types which contribute to prostate morphology. So here on the right, I have them listed. There are groups, or there are subclasses of both epithelial and stromal cells. For epithelial cells, we have two main cell types, which are luminal and basal. Luminal cells that you can see here at the top secrete proteases, or enzymes which cut proteins. And examples of that are PSA and SPINK1. Basal cells, which you can see under luminal cells, maintain the integrity of luminal cells. And beneath luminal cells, you have the stromal cells, which the first of which are fibroblasts. So fibroblasts basically connect tissues together to hold organs in place. The second class is smooth muscle cells, which respond to tissue injury by de-differentiating and differentiating into fibroblasts. So basically, they act as a reservoir for fibroblasts whenever like something bad happens in the prostate organ. And the last group of stromal cells I want to talk about are blood vessel cells, which transports nutrients and oxygen to targets like regions or cells. So I want to emphasize these key points, which I previously talked about. Cell-cell communication is indispensable for prostate organogenesis. And what that means is communication between stromal and epithelial cell is extremely important. It needs to be done in a very controlled fashion in order to make the prostate act in normally. And if this is disrupted by any type of mutagen, whether it's environmental, whether it's chemical, whether it's genetic, can increase one's risk for cancer, for developing cancer. Gene expression is another thing I want to talk about. So gene expression basically connotes that cells have information in the context of DNA, which basically are converted to messenger molecules known as RNA to carry out functions. And that regulation of carrying out functions is known as gene expression. So in the example of the prostate, the androgen receptor controls gene activity of the prostate, since it's the master transcription factor, master molecule, which coordinates normal activity in the prostate. And cancer-causing agents, again, are certain phenomena which increase one's risk for cancer. Okay. So now I want to get into how we study. So now since we've talked about how the prostate is formed and what types of events lead to prostate cancer, the next thing I want to bring up is how do we actually study prostate cancer in the lab? So given that human life is precious, we cannot obviously use human patients to study prostate cancer. However, we can utilize animal models which share a very similar gene expression profile to humans. And the best example of that is our mice. So mice, although like their prostates look so, on the top left, you can see a depiction of the human versus mouse prostate. They look very different. However, the gene expression profiles are similar, and the same types of genetic events lead to cancer in both systems, in both the mice and the humans. So that being said, mice are like an ideal tool for studying prostate cancer. At the bottom here, I want to highlight a novel technique which is being used to map out different cell types in a tissue. This is known as single cell RNA sequencing. And RNA sequencing basically is assessing the levels of gene expression in a given tissue, or the level of the level of gene expression in tissue, in a given tissue. And over here, I hope you can appreciate the different colors and clusters in both of these plots. These colors represent different cell types, and the shared color profile shows that, okay, the same types of cells that constitute a human prostate also constitute a mouse prostate. So hence, this gives a solid rationale to use mice models to study prostate cancer. Okay, so here I have a really big wheel. What does this mean? So this is something that is known widely to cancer research scientists or cancer researchers, and this is known as the hallmark of cancer. And this wheel basically, every segment in this wheel signifies an event, a cancer-associated event. And I'll highlight some of these further in my talk, which are disrupted or changed during prostate cancer, like initiation and progression. Okay. So the first cell type I want to talk about are fibroblasts, which again connect tissues together in an organ. So here on the left, you can see a histological slide of how the healthy prostate looks like. So you can sort of see a nice spatial arrangement of purple to pink, which shows like normal, which is normal, and this looks normal. This is how a normal prostate looks like. Upon prostate cancer onset, the spatial organization is disrupted, and you start to see basically an amorphous like distribution of the cells in this slide. And as prostate cancer progresses, the cells become increasingly de-differentiated, and now there's really no clear like patterns of organization between the cell types, between the different cell types. So now you can see like pink going into purple and purple going into pink compared to the image on the left, which is the healthy prostate. So in the case of fibroblasts, certain genes go up, and these genes are color coded to and correlate with like their overall function. So in the case of fibroblasts, vimentin and alpha SMA, which are adhesion associated genes, which mean genes which help cells to stick together. These genes go up during prostate cancer progression. And another gene called tumor growth factor beta, or TGF beta, also goes up. And TGF beta is a gene associated with pro-inflammation. So the hallmark, connecting this to the hallmark which is significantly affected by fibroblasts like signaling disruption is tumor promoting inflammation. The second cell type, stromal cell type I'm going to talk about is smooth muscle cells. So smooth muscle cells during the progression of prostate cancer demonstrates a few characteristics. One being increased atrophy, or a depletion, or I would say like increased atrophy means basically the cell breaking apart and not looking like it used to. The second characteristic is the degeneration of the basement membrane. And the third characteristic is smooth muscle cells are gradually replaced by cancer-associated fibroblasts. And cancer-associated fibroblasts are fibroblasts which have their signaling disrupted and are more cancer-like. So they proliferate faster and show oncogenic or cancer-related phenotypes. And then in the case of smooth muscle cells, the hallmark which is starkly affected is the one activating invasion and metastasis. So the final cell type I want to talk about are vasculature-associated cells, or blood vessel cells. So during prostate cancer progression, blood vessel cells show increased expression of blood vessel forming genes, such as the vascular endothelial growth factor A protein, or VEGF, which you can see here in red. This protein is upregulated and leads to more blood vessels being formed. And this increases the transport rate of nutrients to the cancer cells or the tumor cells, and will lead to like prostate cancer progression, or prostate cancer growing faster. Another class of proteins which are also affected by prostate cancer progression are matrix metalloproteinases, or MMPs. These proteins actually regulate the basement membrane, and an increase in the expression ends up degrading the basement membrane and leads to invasion of like the blood vessels, as you can see in panels D and E. So looking from panel A through panel E, an increase in VEGF and MMPs degrades this basement membrane right here. As it breaks apart, the blood vessels can extend and stretch to the cancer cells, to reach the cancer cells and provide them nutrients to let them to enable them to grow continuously. Okay. So now that we've talked about how different cell types can be disrupted to cause prostate cancer, I want to tell you guys a little bit about how prostate cancer is actually detected in the clinic. So the first and most widely used test is the PSA blood test. So I brought up PSA before. PSA is actually a protein which is specifically secreted by males, since we have a prostate, and is and its levels are different in a normal person versus a person with cancer. And they are so significantly different that you can use this the levels of PSA in the blood as a diagnostic tool to indicate if someone has prostate cancer, to indicate the risk of someone having prostate cancer. So PSA blood level of less than 4 nanograms per ml means basically connotes a low risk of prostate cancer. A range of 4 to 10 nanograms per ml confers about a 25% risk, and a risk of over 10, a level of over 10 confers a risk of over 50%. These numbers seem arbitrary, and they are not perfect, and I'll explain why in a little bit. They're not perfect because they sort of misrepresent the risk of disease aggressiveness, because they do not account for a lot of plausible reasons why prostate cancer might happen. And one of the biggest causal factors are genetic factors like mutations in genes, and this PSA blood test does not account for that. So it really cannot tell you how aggressive one's prostate cancer can get. So currently, owing to the advent of technology and our improved understanding of the human genome, there are ongoing development of tests and approaches which can help us to detect cancer at an earlier stage, or maybe even before it happens. And one such test is known as the Decipher test, which is a machine learning approach actually used to, and it basically consolidates, assesses the levels of 22 cancer-associated genes, and uses a machine learning approach to predict the risk of prostate cancer aggressiveness. So as you can see in the cartoon right here, the colors of the different silhouettes represents the risk of prostate cancer. So blue represents low risk, red represents high risk, and these rainbow colors are probably just here for fun. But okay. So to sum everything up, we have learned about how the prostate organ is formed from the endoderm of the germ layer. We talked about how different cells in the prostate organ communicate to each other to drive normal homeostasis. They produce and release growth factors such as the androgen receptor and growth factors and the androgen receptor to coordinate activity and function. There are certain factors or events which can make a cell more susceptible to developing cancer, and these come from environmental, chemical, and genetic sources. And a solid understanding of these processes and how they can be disturbed are being currently utilized to improve cancer detection and create better detection approaches such as Decipher. So thank you guys so much for listening to my talk. I'll be happy to take any questions.
Thanks, Jude, so much. So hi, everybody, I'm Jeannie. We have a question so far in the chat. This is a really good question. So Yanira asks if there are certain genes and mutations that denote a high likelihood of developing prostate cancer. So for example, in breast cancer, you can get screened for the BRCA1, BRCA2 gene. Is there any such test for like that for prostate cancer?
Yeah, there are So if I got the question right, are there any like detection approaches for genes like BRCA1 and BRCA2 gene?
Yeah, like are there any specific genes that someone can screen and say, you have an
Right,
X%
right,
chance of developing prostate cancer in the future?
right. Yeah, there are certain gene characteristics, such as there is a fusion event between the two genes, TMPRSS2 and ERG, and the fusion of this increases one's risk for prostate cancer. Another event is also loss in the chromosome 8, in one arm of the chromosome 8, of the chromosome of the 8th chromosome, and specifically at the long arm, which is entitled Q. So if you know a little bit about chromosomes, they're basically like two arms, a short and a long one. The short is known as the P arm, and the long one is known as the Q arm. The Q arm is recurrently deleted in prostate cancer. So those are two examples of genetic events that you can look for in prostate cancer.
And so could this be detected at an earlier age? So just to keep going with this this sort of like parallel, there's a lot of women who are given a risk score, and they can do things like,
Right.
preventative mastectomies, because of, just to avoid breast cancer altogether.
Mhm.
Are we there yet with our understanding of prostate cancer?
No, I wouldn't say there's any like preventative avenues being used for prostate cancer. If anything, I would say like the early detection approaches are currently being optimized. One of the biggest problems with prostate cancer is actually over treatment of the disease, because metrics like detection approaches such as PSA sort of overrepresent how serious prostate cancer is. And it's actually, it's the truth of prostate cancer is it's not that lethal in the first 5 years of having the disease. Patients actually have like over 90% risk survival rates. And after then, their survival only gets worse due to treatment emergent genetic like aberrations. So, and that means mutations which happen as a result of therapy. So these are like resistance mechanisms, essentially. So the short answer is no, we don't have any preventative methods just yet.
Yeah, it's fascinating how our cells can behave so differently in different cancers, and just where we are with some cancers in terms of our understanding versus others, but thank you for that answer. So we have another question from Vivian. And they ask, in the presentation, there was a part that showed differences in the amounts of cell types between the mouse and the human prostate. So I think that they're referring to that, the diagram of the mouse versus human. Is there a specific reason why there would be varying amounts? Oh, I guess this would also be the plots that you showed.
Right, so the single cell RNA sequencing plots. Yes, I would say there's no specific reason besides it's just owing to anatomical differences between these two species. The actual amount of the different cell types between these two species does not really mean anything. It doesn't have any connotation, any significance, I mean.
So Eugia, and I apologize if I'm not pronouncing your name correctly, asks, why do cancerous cells produce more PSA?
Oh, so PSA is a gene regulated by the androgen receptor. So as I said in my talk, the androgen receptor is the master molecule for prostate normal function. And when the prostate experiences an event which makes it cancerous, the androgen receptor like activity is like shoots up. So every gene which is regulated by the androgen receptor also shoots up, and PSA is just one example of that.
Cool. I think we're gonna just stop the sharing of the slides just so we can, yeah, there we go. Awesome, thank you. Okay. So Mehmet asks an interesting question about growth hormones. So I know that people are provided some sort of hormone therapy. Some people are administered growth hormones for varying conditions. Does using these types of hormone therapies make prostate cells more susceptible for cancer compared to other types of cells?
Yes, 100%. So just following up from my previous comment, the issue with prostate cancer treatment is it's the detection approaches, like I said, overrepresent the disease. And the standard line of therapy for prostate cancer is actually to remove androgens, or the source of androgens, or testosterone. And that actually leads to mutations in genes which bypass the androgen receptor like signaling axis and lead to a state of prostate cancer which doesn't respond to androgen levels. And that state is known as castration-resistant prostate cancer. So the answer to your question is treating prostate cancer actually makes it worse. It actually makes it worse, yes. And this necessitates the need for better detection approaches. So we can be better informed as to like what type of treatment to give, because there are some patients who don't need the androgen taken away, but like that is the standard line of therapy for all patients who have localized advanced prostate cancer, really.
So I think that really leads in well to Mehmet's second question around the machine learning algorithm for prostate cancer detection. How confident can we be in these types of detection systems?
It's definitely a work in progress. I would say like, well, that's where machine learning comes in. So machine learning is great in the sense that it uses many variables, like it's sort of a multivariate avenue for predicting like risk of prostate cancer. So it's not just going to look at like, okay, like PSA blood levels. It's going to look at PSA blood levels. It's going to look at the histological status of the cells. It's going to look at other like gene mutations and basically like consider all these different factors and assign a risk score. So in that sense, using an approach like such as machine learning would yield a more accurate diagnosis of a patient, right? Rather than using like just one metric, like such as the PSA or such as maybe a biopsy.
Yeah, it seems like there's just so many different ways that we can consider like gathering data for detection and diagnosis. And so this really plays so well into the theme of how do we study ourselves. And it sounds like through multiple routes is a really prominent answer. How about a question from Sumama so I know you showed earlier in your talk about dedifferentiated cell types in the progression of prostate cancer. So why do cancerous prostate why do cells that are, generating cancerous tissue dedifferentiate? And does that population of dedifferentiated cells impact normal prostate function?
Okay, that's an awesome question. So the undifferentiation of prostate tissue in cancer is not just a prostate cancer specific event. It's actually an event which is common to all cancers. So cancer in the most basic terms means uncontrolled cell division, right? Because we do have like gene expression rules which like coordinates normal activity, and once like these rules are like disrupted, there's cancer. And what happens is once cancer starts to happen, cells which have committed to a certain like job. So for example, luminal cells, which have committed to secreting molecules, stop doing that. And then they become stem cells. So basically they undifferentiate and become cells which can basically continue to grow, never die, and just lead to tumor formation. So that's really the, the hallmark, like one of the most important events in all types of cancers. in any, you can name your cancer of choice. I would say like in breast cancer, the same thing happens. Breast mammary cells de-differentiate into stem cells and continue to proliferate and grow, and that leads to cancer progression.
These are some really great questions. Okay, we'll take one from Samuel. If a prostate cancer could be detected early, could medications be used to prevent angiogenesis from occurring, which could inhibit the growth of the tumor? And would it possibly damage other organs that depend on angiogenesis for their own function? So I guess this gets at sort of that, you know, how do we kill tumor cells without impacting self and healthy cells?
Right. That's a great question.
If you knew that question, the answer to that question, I feel like that would be sort of like a cure to cancer, right? But yeah, like what are your thoughts anyway on moving that forward?
Oh yeah, great question. It makes me ponder. I would say off the top of my head, I don't know if there's any like angiogenic inhibitors or drugs which like inhibits the process of angiogenesis or blood vessel formation. However, I would say like if we can like formulate drugs which target I would say cancer associated vasculature cells versus like normal, so like target them specifically and don't target like normal blood vessel cells, that would be a great avenue to treat prostate cancer and all cancers actually.
Cool. So you also mentioned at the top of your talk carcinogens. Is there a particular carcinogen? So this question is from Rui. Is there a particular carcinogen that might lead to more prostate cancer development, for example? Or are carcinogens sort of like they'll cause cancer and whatever happens for the type of cancer?
Right. I do not think there are any prostate cancer specific like carcinogens. No, there aren't. But that's a great question. I will check again. To take that with a grain of salt, I will check again, but great question.
Okay, so there's another, there's quite a long sort of thought process here from, and please forgive me if I'm pronouncing your name incorrectly, from Runji. So they bring up P53 and how that's the most common cancer mutation.
Mm-hmm.
How did that happen? So how did P53 become like the oncogene? If you even sort of are familiar with how that might have even evolved.
Right.
Yeah.
So P53 is the reason why it's the most commonly mutated gene in cancers is because it has it's very indispensable for the cell cycle actually, and the cell cycle is basically a series of stages through which a cell goes to divide from one cell to two cells, basically, or to replicate. And P53 is important for the first parts of like that cell cycle process, known as like the G1 to the S stage, really. So the fact that it's indispensable for that first step gives reason to why it's so commonly mutated. So what happens is it's actually the specific mutation with P53 is a loss. It's a homozygous loss. Like homozygous means both copies of like P53 are lost, and that leads to like the brakes on the cell cycle. That leads to the brakes being taken off like the cell cycle control mechanisms, and leads to cell cycle happening much faster. I know, I should rather say in a more uncontrolled fashion, the cell cycle happening in a more uncontrolled fashion, and eventually ultimately leads to cancer.
Wow.
In this context, I hope I answered your question.
So master regulator, right?
Yeah, it's actually called the guardian of the genome.
Okay.
Yeah, that's what people call it. Yeah. That's what scientists call it, the guardian of the genome. Cool. Because that's what it really is.
It's a marvel. It's a marvel gene. I think Guardians are Marvel, or are they DC? I never remember the comic. Sorry.
They're Marvel.
They're Marvel. Okay. Yeah. I should know this by now. All right. We have one last question. So Mehmet asks another fantastic question around how prostate cancer, maybe this could be specified into prostate cancer cell types. How do they act in later stages during metastasis? And does prostate cancer sort of cell types in late stage, are they different compared to other late stage cancer cell types? And are there different approaches for treatment?
Yes, so this is an emerging field actually. So along the line of metastasis, we can classify prostate cancer into certain groups. The different prostate cancer cells. There are cells which still respond to androgen treatment, and those are known as androgen. That's known as androgen sensitive prostate cancer, right? There are cells which don't respond to androgen treatment, so they have mutations which bypass androgen receptor signaling. These are known as androgen independent or androgen insensitive prostate cancer. And the third type is cells which don't respond to like either like androgen they don't care about androgens in any fashion. And their genetic the event which coordinates like their growth is still being uncovered. So that third class is known as double negative prostate cancer, DN prostate cancer. So these are the three groups which are have been accepted by the field. And DN prostate cancer is the least studied. That actually involves like a multitude of different cancer drivers, such as NMYC, which is a neural lineage gene, AP1, which I actually study for my thesis work, and others.
Wow. And so I guess I have one actually, I would like to ask one last question. So I know you mentioned that there's different classes of prostate cancer. And I have to say, I am more familiar, at least at surface level, with sort of the ways that breast cancer impacts different populations, just because as a woman, I have had more access and more sort of, I've read a little bit more about breast cancer because it's more related to women. And so I know, for example, like triple negative breast cancer impacts women who have African ancestry more than any other ethnic group. Does prostate cancer, the different classes of prostate cancer, are they more enriched in a particular population? And should that impact how we think about studying and treating those classes of prostate cancer?
Right. This is an awesome question. Yes, there are. The short answer is there are racial disparities in, I would say, hormone-driven cancers like breast cancer and prostate cancer. The reasons for that are still being debated. I would say some people think it's more genetic, some people think it's more socioeconomic. And depending on who you talk to, you might get like one answer or the other. But I would say this is okay. I would say that one of the most compelling genes which are actually enriched in one demographic is a gene called CHD1. It's more lost in African, like in males of African ancestry than males of any other like demographic. However, that's only like one example. That's only one example. We still need to like uncover more to like, okay, like conclude that the difference in racial disparities in prostate cancer incidence and mortality are due to genetic factors and not like socioeconomic factors. But then again, it's being debated. Some people really think like, okay, the reason why different groups of people have different prevalence rates is because of like things like access to health, the type of diet, where you live, if you live in like in a very polluted area. So things like that, of course, like increase like your risk of any type of cancer and prostate cancer. Yeah.